I will turn it over to Dr. Mark McMills.
Thank you very much. I decided to kind of take a different tact here. This isn't a usual talk that we give. I'm going to talk a little bit about my work at the very end. But what I decided to do was to look at drug discovery in the United States mostly and how it has progressed. And essentially what I'd like to do is look at the different kinds of procedures in terms of drug discovery. And so we're going to look at ostensibly 3 different drugs that you could realistically say have changed the world. One is going to be penicillin. The other is Taxall. And the final one is it's a protease inhibitor for the HIV virus. So easily you could say these things have really changed the way things occur and help literally hundreds of thousands to millions of people. And so I think understanding some of this before the talk tonight, somebody in the media was asking me a question about that. And I think it is important that all of us understand a reasonable amount about drug discovery and what is it that's being done to find agents that are going to go into our bodies to be used as medicinal agents for different types of things in terms of whether it's cancer, whether it's diabetes, whether all these different things that are out there. So I thought we'll take a little bit different tact tonight and see how it goes. And what I'm going to do is I'll tell stories about each one of these different ones. The one that will be a bit contentious, and actually I want to get some feedback from you guys, is when I talk about Taxall. Taxall has some very interesting things that go with it, so we'll see where that gets us to. All right, so if we think about this, we really want to know why are we even doing drug discovery? What's the purpose? And if we think about it, what we're really trying to do is interact with various systems and signaling pathways and things in a human body that determine what goes on with us, that will determine whether we're healthy, whether we're diseased, whether all these different things. Because even as much as we attack these with different kinds of drugs and things like that, other things are going on, that their cancer is attacking various things in your body. All these other kinds of disease states, they're also doing the same sorts of things. So the best thing to do is to understand a little bit about why that goes on. So I've sort of given you a way to think about this a little bit, but just take this as a grain of salt as we start going through this. So in the first part of the talk, what I'd like to do is tell you a little bit about the drug discovery process. Now, hopefully most of you have heard of all these different things in terms of phase 1, phase 2, phase 3 clinical trials, things like that. And we'll talk about that a little bit just to get a sense of what goes on with these. And so I think this next one is kind of an interesting, and I apologize for sitting down in front of you, but I've got some issues with the back that are a problem. So I apologize. But all right, so we want to talk about what goes on here, and especially in terms of timelines and things like that. And so as we look at it, the first majority of what's going on will be the discovery of a molecule, the synthesis, purification, testing, and then some institutional, like OU has an institutional review board that if we have drugs that are going out, things like that, they take a look at what's going on, especially if we're doing testing. Animal testing has to be approved by the university, so it's not something that's taken lightly. And so everybody is concerned about it when we look at it. Now this is a big, huge part of this. This can be a long part of the process in terms of bringing a drug to market. Tends to be about 6.5 to 7 years overall, but it can be upwards toward 11, so it's quite a number. Once you finish with all of this, then the key is to hit this point where you're doing what's known as an IND, an investigational new drug application to the FDA. And this is a big deal. This takes a huge amount of energy. I think, where's Rox? There's Rox. Rox, I think, is one of the few people in this place that's actually brought INDs to the FDA and gotten them through. So she actually knows how difficult this actually is. So it is one of the things that, especially if you have a company or you're a faculty member who has something that they're bringing, it's something you have to learn about fairly quickly. Then you go through phase 1, phase 2, and phase 3. We'll talk about those in a second in terms of what those require. And then once you get through this, think about how far you're along in this process. At this point, once you've done this, this is where you're likely to patent, right, when you come up with a synthesis and find something that's interesting. And so you figure that we have, what, is it 17 or 19 years on patent now? 17 still? Okay. So the government gives us 17 years on patent. And the problem is, we get through a lot of that just getting through the process of trying to bring a drug to market. And so if you look at it from the standpoint of a drug company, they're upset because they will lose patent protection on that fairly quickly once they get through all of this different strategies in terms of the drug discovery. And so there's always the give and take, though, that some of these drugs, as you may have seen, are expensive. They're not very available. You have to take them, a number of them, all sorts of things that go with that. So there's a push and shove between the drug companies and the people at large in terms of how you're going to get them, how much you're going to pay for them, all these other things. But as I say down here, one of the problems of this is that we're probably talking about $1 billion investment from a drug company into a drug. Now, let me ask some questions here. In terms of a drug that we're bringing to market and we're spending $1 billion, is that all spent, let's see, I'll use the word wisely. Is it spent by a drug company to do exactly what they need to do to bring a drug to market? What do you think? Why? The answer was no. Why?
We've got between the cost of the patent and all the man hours that have to be put into it.
Well, but okay, and that's not unreasonable. But what you're talking about is the fact that is all of that $1 billion spent directly bringing that drug to market? And so part of that answer is yes, but there's a part of it. Okay, think about one of the things to ask any of you ask your doctor is ask them if they've been on any trips from reps, from pharmaceutical reps, and see if they should be able to tell you whether they've been on these trips. A lot of times they're doing what's known as continuing education to somewhere like Bora Bora. Now, one may think that's not a particularly good use, but that ends up being part of the cost of bringing the drug to market. And so a number of people would say that's not really a reasonable way to spend that makes this cost so prohibitive in terms of the overall cost of a drug. So those are some of the things you have to think about as these things are being processed through all this. Okay, so then the last two parts are the FDA review, which can take up to about 2 years. This is the one that's a problem. Once all the information's in, FDA will take time to review all the data. They go through all the process. They look at syntheses. They look at the manufacturing. They look at all the different things that are associated with that. And so this is a fairly tough deal for them. And for the drug companies, these three are the real problematic ones because anytime during these three phases, a drug can crap out. It can find you have toxicity. It has something that there are issues with lung, any kind of organ, and this will cause problems. And they look at multiple types of testing. So they will test on mice. They will test on rats. They will test on dogs. They will test on various kinds of animals. And so at any point, this can become a problem. And think about it, if a drug company gets to phase 3 and suddenly they see, say, liver toxicity showing up, then that drug is done most likely. And they've got to, somehow they have to look and see if there's a second generation that they can use for that. So this is a really tough deal for the companies, and it is a tough time for them. So that's one of the things to think about as we do this. And again, it's a huge monetary investment. Okay, so then just to show you what goes through here, if you look in this process that we're looking at, phase 1, 2, 3, and then all the prior stuff, we start with something on the order of 5 to 10,000 compounds. At the end of that whole process, we're talking about likely one approved drug. So these companies that do this is a huge expense for them because they have to bring all sorts of drugs through this process. And so it becomes very expensive, it becomes painful for them. The researchers that do the work, they put their life's blood and sweat into it. And so at the end of the day, it could be somebody comes in on a Friday and says, the drug didn't make it through, we're done, and you are completely changing what your job was, what kind of things you were making. So this becomes a very hard process for this. Okay, one other thing I wanted to do then is show you in this process, we're talking about a 12 to 15 year process time limit. And so I have an example that was brought up from 2007. And what I wanted to show you is all the different things that were happening 15 years when they actually started the process of bringing that drug to market. And so think about this in 2007. So the world events that were out there is the Yugoslavia collapsed, then Slovenia and Croatia come in. Aladdin was the top-selling movie. Home Alone 2 and Batman Returns. How many of you have even seen those? Okay, very good. Jay Leno, well, at least we don't have Jay Leno now. So at that point, we still had Jay Leno. Ted Turner, which started CNN. So these are things that are great. I mean, does anybody actually remember Andre Agassi as a tennis player? All right, she does. Okay, so it is a huge time frame that we're talking about overall that this is happening. So just keep that as a sense in your mind. But there are trade-offs that we're talking about in that, yes, the drug companies put a huge amount into it, but there can be drugs out there that are necessary for somebody who is very sick that could be $10,000 a dose. Those kind of things become very, very hard for a person to deal with, unless they have pretty good insurance, which it tends not to be that good. So it can be an expensive process. All right, so in the phase 1, 2, and 3, we want to look at this. Phase 1 clinical trials take about 20 to 100 healthy people. These are healthy individuals who do not have whatever the disease state is that they're looking at. They want to see, is this drug going to cause any issues with somebody who is healthy? So this is where they're going to look for liver toxicity. They're going to look for any other toxicity that's out there or any other problems. And it gives them a chance to look at all sorts of things in terms of metabolism, pharmacodynamics, anything that happens. They start to get a sense of what's going on with the drug. So this is one way of getting an idea of what's going on with that drug and what can happen with it. In terms of phase 2 then, we start to move into much larger. So we're now at about 100 to 200. And now we're starting to deal with people that have the disease state that we have to deal with. So these can be people that are fairly sick, and we want to see what happens to them. And one of the good things that goes on with the FDA is that if they notice in trials that a drug is particularly effective, they may actually stop the trial and give people the drug itself. Because what they'll do is they'll do double blinds and placebo tests to make sure that the drugs that they're looking at are reasonable. And so for the most part, they will go through this and be able to get to some idea, a question about what's going on with this. And again, the researchers get a huge amount of information from each one of these tests. And this one, dosing is important, and then scheduling for the drug. So these are all important. And then again, you have to go finish these things to move on to the next trial. And then finally, we hit phase 3. This one is the one that's really, really expensive. We're talking about anywhere between 1,000 and 5,000 patients. These will be at generally hospitals all over the country. And it is a huge amount of money. What you see is, in a number of cases, drug companies and various other people that do these tests will go to NIH, things like that, to try and get money to run these studies because they are so expensive. But for the most part, it becomes part of the $1 billion that we're looking at in terms of overall cost. These are long. These can take up to 3 years. It's a huge study with lots of people and takes a lot to coordinate. So it is a massive undertaking for this. But this is where they have to get through all of the things that go through this. And we have to have some idea of what's going on with it. Okay, so that's sort of the drug approval process, I guess. The FDA uses this for almost all of the drugs that are out there, the things that are going to be given to humans. So we usually look at these and go with that. And so again, I wanted to go back and look at 3 different types of drugs, and especially I chose 3 different ones because of the way that they were discovered. The first one that we're going to talk about is penicillin G, well, penicillin. And this I'm calling serendipity, and we'll see that why in a minute. The second one is Taxall, and Taxall is a natural product that was developed that way. And finally, this one is a Darunavir, and this is a protease inhibitor. And I call this rational drug discovery because what they had was an idea of the enzyme that's part of the HIV protease system. They knew what it looked like, and so what they did was model the compound to that. And so it gave them an idea of what's going on. And then finally, I'll give a real short spiel about some of the drug efforts in my lab. And we work on something called 4-ball. And so I'll talk about that a little bit. So just to lead things off then, here would be what happens with penicillin. This is a picture from back in 1948 where a mother is actually giving her child a shot, which sort of amazed me. I didn't know they actually did that even back then. But I can't imagine. How many of you are mothers here that want to start sticking your kids with needles? I bet that's not a huge number of you. So, all right. So, in terms of penicillin, it was an interesting molecule. It was discovered by Alexander Fleming. And the two pictures here are Fleming in his lab. He was in England, but he's actually a Scotsman. And so he came back at the end of the summer from a vacation in Scotland. And as is typical, he came into his lab and found out it looked like most of our labs do, a giant, horrible-looking piece of junk where stuff's everywhere. And I think he came back and decided he needed to clean things up. And so he had Petri dishes everywhere in his lab and started to say, okay, I need to take these and put them in places. And I think he had them actually so they were different types of strains of bacteria that he was looking at. And so he got into one group that we know now as Staph aureus. And he was looking at those, and he actually picked up this one and found that he had a mold growing in, and that was this. This mold was growing in the plate as well as all of the bacteria that's there. But what was really interesting to him was he said, well, wait a second. There's something really special about this, that there's this area around the mold that is a kill zone, that none of the bacteria is growing in this area. And so that it dawned on him that something was very special about that. And he was able to take these and move it on to some other things and look at some of the things that were out there. The problem is that St. Mary's in England, where his lab was, is a very small place. It didn't have very much help in terms of chemistry background. It didn't have the labs to do the things. And so what he did was chose a guy named Florey at Oxford, who was the one who was going to do, he was a pathologist. And so he was the one that was chosen to move this on in terms of figuring out what's going on. Fleming kept doing a lot of research with this, but because this had to move into bigger and better things, it had to move into a place that had an easier way of doing this. Particularly the chemists, the biologists needed to move this out of a simple lab and be able to take it out. And so one of the things you can think about is, here's our mold. We have to be able to find out what is the element in that that's toxic to the bacteria that's there. And so part of this requires you to isolate. It requires you to figure out what it is that's there and what we can use it against. And so in many of these cases, Florey found all sorts of different things to put it against. He found a lot of things that were happening with the drug and against other types of bacteria that were possible to use it against. Now, one of the things that happened at this point is that as they were looking at this, as you see in this, it was 1928, but Florey decided that he actually needed to come to the United States and find out about drug companies in the United States, helping them to move this forward. So why would Florey have chosen the United States to move this process to? What is it at that time that you had to think about that was going on? Say again? War. Okay, very good. World War II was happening, and most of the resources by England were being put toward their war effort. They did not have anything to do to bring this forward. And one of the really important things about a compound like penicillin is that there were many, many people in the war getting incredibly sick. All it would take is one small nick, and they would get infections that would kill them in the space of 2 weeks. And so these infections were incredibly bad, and they had to figure out a way to get through this. So this became part of the war effort for both the United States and for England. But one of the things is that there were drug companies in the United States that were set up to start bringing this forward and to do the isolations, to figure out structure, to do all the other things that were associated with this. And so one of the things that happened is they looked at drug companies like Bristol Myers Squibb, Pfizer, were two of the main ones. And so if you look, here's some pictures from it. They're a little out of. These are the fermenters that you see. This one is at Merck in 1945. These are some of the deep tanks at Pfizer. And then this is at Bristol Myers Squibb. They started putting things together. Sorry, the picture is pretty bad in that. But this is where they started fermenting things. And then this is an old cover of the American Pharmaceutical Association when penicillin first came around. And it started being very important to these bacterial infections that people had. And then being a chemistry professor, I couldn't let it go, but I did have to show you that in 1957, the penicillins were made strictly chemically. And so you can see a chemist named Sheehan put this together. And I'm trying to think, how many? We've got one from my class. Anyone else from my organic class? There we go. Okay. What does that look like? You guys remember? Let him off the hook? Okay. Remember the Gabriel synthesis? That's for the Gabriel synthesis. So this is actually something, despite seeing it just sort of ad hoc in my class, it actually is something that's been used and is used today in a lot of different things. So I'm not going to go through the gory details of it, but suffice it to say that Sheehan was able to put these things together. Although if you look, some of the yields here, there was another one, the last yield, unfortunately, these are not such great yields, and this causes problems. And I'll actually talk to you about that a little bit later on. But they're able to get this thing down. They get this compound that they put together to get the penicillin structure for this. So they are able to put this together. And if you look, it's basically 1, 2, 3, 4, 5 steps or so. And this is not an unreasonable industrial reaction that we can do. If it's more than that, it starts to get to be problematic in terms of how we do them. So I want you to take away that penicillin was a sort of not something that he planned on, but it showed himself in the lab, and it was something that that Fleming was smart enough to pick up on and be able to use. And so this, for all of you that are budding scientists, this is an important part of things that go on in the lab. You need to keep your eye out on things that are going on. A side reaction may actually be important, may actually be interesting to you, even though it's not really what you're trying to do. It may be the next coming of penicillin. So keep your eyes out and make sure you understand the things that are going on around you. Okay, we'll shift to the second one then. And this is going to be this natural product called Taxall. And one thing I do want you to take away is that one of the unfortunate things that's happened in the United States and all over the world is that many of the drug companies that are many of the drug companies that are out there have decided to let their natural products groups go. They don't keep up with them, which is really unfortunate. because the majority of the drugs that have come through that are actual drugs that are being used in humans today came from some natural product source. But what we find is that this is a very expensive way to do it. But what we find is that nature brings us an incredible diversity of structure that in a lot of ways we don't think of, that we may not see, that are very, very important to what we do. And so my plug is that natural products are an important way for us to come up with new drugs every day. And so it is something that I do hope that many of these drug companies will go back to. Several have. And so I think as a chemist, myself and my colleagues applaud that. So what I want to show you here is the Taxall molecule looks like this. Now this is a fairly different type of structure, and it's one that took quite a bit to bring to the chemist's attention. And I'll tell you about that in a little bit. But the main thing here is this 8-member ring. It has a 6-member ring on it, and then it has this nonlinear fused 6-member ring too. And so this is kind of an odd-looking structure. And when I first saw this, when I was an undergraduate, it was all of us looked at it and went, "Whoa, nobody's going to make that." And it took quite a while for it to be chemically produced. So we'll come back to that a bit. Okay, so this Taxall, this is what the Taxus brevifolio looks like, the structure of the thing that brings it forward. And these come from old-growth Pacific yews that are found in this area in Canada and the United States. And basically, the Pacific yew is this this bark that comes off of it. And we're going to talk a little bit about that. So these are the keys for what these things look like. And the initial studies were done by a guy named Arthur Barkley. Arthur Barkley was a botanist who was contracted through the Department of Agriculture and worked for the National Cancer Institute as well. And so he actually, his job was to go out in the wild and bring back all sorts of stems and roots and bark and various things from different kinds of structures to see if there was anything in them that we find interesting. And so he was the one who did all the initial work on bringing it in as the bark and all the other stuff that comes with it. And then once he had finished with his work in it, in 1967, the NCI assigned the extracts of this Taxus brevifolia to these two men. This is Mensa Wani and Monroe Wall. And they were contractors for the NCI through the Research Triangle Institute in North Carolina. They did the lion's share of the work in terms of isolation and structure elucidation over about a 4-year period. And so from them, they're the ones that we actually know what the Taxall structure looked like and what was important about it. Okay, so now as they brought this forward from 1964 on to 1967 and then further, what we found was that we're starting to get more and more interest in Taxall itself. It's starting to show interesting things in terms of activity against various types of cancers, and it has some activities that we hadn't seen previously in various things. And so initially it showed good activity against leukemia, carcinosarcomas, and mouse melanoma models that were out there. And so it piqued curiosity of a bunch of researchers. Now, this is the really important part of this, and that is that it has extremely limited water solubility. Okay, and so that becomes a problem for us because, well, what do I mean by this? Well, I can't give it if there's very little solubility. I can't give this as an oral drug. If I can't give it as an oral drug, it's much harder to get into people. We have to use needles to stick bags into them, so we get solvents and various things into them with the compound. And so it's a much harder way to get a drug into a person. And so that caused a lot of concern out there. But even more so was this limited availability in quantity. And that is, I'll show you that coming up in a second in terms of what we want to look at. So hold that for a second. In 1979, this became really important, though, and that is Susan Horowitz at Yale Med School found out that Taxall worked by a completely different way than other drugs. If you look, we're essentially looking, many of you are biology majors, right? Yes? No? Okay. So you know what happens when a cell divides, right? It forms spindles and it pulls apart. Yes? Okay, well the spindle that forms, this is really important in terms of the overall way that this goes. And we know of, as chemists, a bunch of poisons that will work on that. There are a bunch of indole alkaloids such as vincristine, vinblastine, that are spindle poisons. And so they will stop the assembly of microtubules and stop this thing from pulling apart. Taxall was a really interesting thing in that what it did was the opposite. It actually caused the assembly of these microtubules, but it wouldn't let them disassemble. And so it was a totally different mechanism in terms of attack of looking for a drug that we could intercept this. And so Susan Horowitz was the first one to find this. And so this sort of is the simple way of seeing what happens with this. And that's, there are two tubulin parts, alpha and beta. And if you look, the normal way they will go together and start forming these things into the microtubules. And these go back and forth. So they'll form and then they'll break apart. And that's the typical way that a cell cycle will go. In this case, though, what we see is Taxall promotes this formation, but then it can't go away from this. Once this forms, it's done. And that's why you see the big arrow going that way. So this was the way that we could intercept this and find out a way to the drug would stop it from doing anything. And so this was a really interesting way of looking at this. So let me show you, this is generally what we consider the microtubules to look like. Obviously, this is just a cartoon of it, but it looks essentially something like that. We have done X-ray crystallography work, and this is where we see the alpha tubulin is here, the beta tubulin is here, and if you look, this molecule right here is the Taxall that's bound to it. And then we can see in that part of it, the Taxall bound within this, and this is what it will look like. Now, obviously, these are cartoonish looking things. These are ribbons and various things that denote huge numbers of atoms and molecules that are part of this overall structure. So these have to fit in a very specific way. And Taxall has been found to bind very, very specifically to the beta tubulin. And we've gone further than that. There are a bunch of other drugs now that will do the same kind of things. But for the most part, Taxall does that specifically. Okay, so now you guys have listened to me blab on about a bunch of things. But does a molecule like Taxall actually work? And that's obviously going to be the bread and pudding of this whole thing. And what I found is that if you look at what's going on here, this is a human organ. And what they found was that it's a little bit hard to see them. There's a tumor here, a tumor here, a tumor there. There's another one here, and another one there, and there. This was the first time. This is after then. They gave a regimen of Taxall. If you look, essentially all of the tumors are gone, all of them. And this excited people immensely, especially in terms of ovarian cancer and breast cancer. These are two really horrible cancers that women get, and for the most part, and they are extremely tough for people. And so finding something like Taxall has been an incredible boon to this taking care of these parts of the disease states. So these are really, really important. And so I just wanted you to see that these things actually work. They are important, and it is something that has changed a huge number of lives. Okay, so now remember I talked a little bit about this supply issue. And I want to talk about that because this is actually a really, really important thing. And that is, okay, Wani and Wall, those two guys that were working in North Carolina, they found that the average yew tree would give up about 2 kilograms of dried bark. Out of that dried bark, 10 kilograms would provide 1 gram of Taxall. That's not very much. That won't even take care of one person in terms of the overall amount they need for a total regimen to go through the drug system. So then if we look in this isolation process that we're looking at, again, they're going into these northwest forests. These are old growth yew trees. They've been there for hundreds, if not thousands of years. And they're basically taking the bark off, which does what? You take off the bark, the tree is dead. So look at the numbers, though. 1974 to 1989, we're talking about approximately 7,500 trees and about 6,500 pounds of bark. It's not that much, but then look, in 1987, Taxall went into phase 2 trials. And now NCI wants, the National Cancer Institute, wants 60,000 pounds of bark. This is now for both 1987 and 1988. This is a huge amount of trees that they're going to kill. And they're 1990s because now it's passed through phase 2, it's in phase 3. Now they need 130 kilograms of Taxall, which they estimate to be about 1.5 million pounds of bark. You have just decimated a forest completely if they do that. And so there were a huge number of issues that came up with this. There were a number of environmental groups in Oregon and Washington that said, wait a second, you can't kill this whole forest to save people. But then the other side is, if this is your mom or dad who's dying, what are you going to say? Tear down the tree, no problem. So there was a huge amount of fighting going on in the early 90s about this. And so, go ahead. Somebody said something. So, to go on with that then, they said, the people at NCI and all over the American Cancer Society and chemists in general said, we can't do this. We cannot tear down this whole forest to get the amount that we need. We'll never be able to get it because, think about it, essentially if we destroy that forest, we're done. All of the Taxall that's available to us is gone because it takes years and years and years for these trees to grow the bark that we need for this. So this becomes an issue. So the way that we decided to approach this is instead our supply then would come from either semi-synthesis, and I'm going to show you that in a second, cell culture fermentation, or chemical synthesis. Those were the three possible ways to approach this that were a much more reasonable pathway to get the amount that we need. Okay? And so the first one is semi-synthesis. And now this semi-synthesis comes from this compound, which is called 3-deacetyl-baccatin. And if you look at the compound here, this looks pretty similar to what we saw previously for Taxall, except for what? Does anybody remember? It's off of this OH. There was a side chain that ended up off of that. Okay? And so what they decided was they found that they could, in this Taxus Baccata, this is what it looks like. And actually, I think if you look in a number of yards, these are ornamental type of yew plants that are out there. You can actually find them around. And what they found was the reason this was interesting to us is because now, in terms of semi-synthesis, we get a bunch of this out, but it comes not from the bark, it comes from these needles. These needles will grow back. So we can harvest trees and not kill them. And we still get the compound that we need for the semi-synthesis. And so this will, these kind of plants then produce a substantial amount of this. Now, that's of interest to us because if you see this picture of the guy here, this is Bob Holton. Bob Holton is the one who patented the way to put on the side chain here. Okay? Now, how many of you know who John Kopchik is? Okay. John has made a fair amount of money for himself and OU, correct? He generated a drug for OU, and that's gone through. Well, okay, so Bob Holton developed this side chain for Taxall. Bob Holton and Florida State, where he is a professor, made $351 million from the sale of that compound. That is a little bit more than John made. John would love to have $351 million. Okay, but I want just to give you one interesting story about Professor Holton. Professor Holton had lots and lots of money to spend. And he has started a company and started a number of things. But he also has decided that he is sort of in control of the chemistry department at Florida State and decided that the university needed to build a building for them. But he put his money where his mouth is. He gave the university I think somewhere on the order of $39 million from his funds and from the amount that the university has in conjunction with him. And basically came to them and said, look, I will give you this money, you will build the building, you will also fund some faculty positions. And the first person that he dealt with, the president at Florida State, was okay with that. About midway through, before they started developing the new building, a new president came in, and he decided that that wasn't a really good use of funds from Florida State. He said, we'll build the building, but we're not going to fund the professorships, and we're not going to build the building the way that you want it. And so there was a huge fight that went on between these two. And finally, in about 2006, Holton actually took Florida State to court. And he partially won, where he got back his donation from that he was giving toward the building, but Florida State maintained control over the joint money that they had, and they can use that for anything they want. So this was sort of an interesting sidelight that these things go on, and obviously, and I'm sure you've never seen this before from any of your professors, that we have egos, right? But one could say that Professor Holton may have a bit of an ego, just a bit. So that's the story of what goes on with that. All right, so the second in terms of the supply chain was the cell fermentation. And I'm not going to go over this, but I just want to show you that actually this is the Taxall biosynthesis pathway to go from these geranyl pyrophosphates all the way to Taxall. And so these are, if you look, there's a bunch of different enzymes that are there, and we can get through this whole process. This now is done in cell culture. The majority of the Taxall that's available in the United States actually comes from cell culture and semi-synthesis. And so they've been able to work out methods to get this to grow and to do all the things that they need with it. So this Taxall generally comes from something like this cell culture. Okay, but the last one that was there was chemical synthesis, correct? And me being a synthetic chemist, we need to address that a little bit. And so what I wanted to show you is that here are some of the different syntheses by some of the bigger names in synthetic chemistry. You go from K.C. Nicolau in 1994, starting from this simple compound. Nicolau was the first synthesis, along with Holton. They were essentially together. And Holton came from a patchouli. So these are interesting. The one that gave the most, the best synthesis is actually by Paul Wender in 1997. But look at what this is. It's 37 steps. And if you look here for that 37 steps, the overall yield was 0.4%. That is a really, really small yield. It is impossible to get the kind of amount we need of Taxall through a synthetic route. And I've shown that here. So if you have 25 linear steps with 25 purification steps at 80% efficiency, essentially 1 kilogram of starting material will produce 14 milligrams of Taxall. This is not a reasonable way of doing this in terms of getting us the amount that we need. And so for a 25-step process, it will require 71,000 tons of starting material to get to the kind of Taxall that we need to give to all the patients. So it became readily apparent that chemical synthesis was not a reasonable pathway for this. And so that's why the fermentation and the semi-synthesis have become our major source of this. Okay, just to show you the last part of this, and medicinal chemists out there have done an incredible amount of work on Taxall. And if you look, all of these different places that I've put these different little things, these are all areas that chemists, medicinal chemists, and synthetic chemists have changed the things that are there and found whether it improves the drug or makes it worse. And so they've gone all these things, all through these different things to make literally thousands of different kinds of compounds and find out what gives us the best activity out of all of those. Okay? All right, so one last one I want to talk about is the change to this, these protease inhibitors. And so just to give you a sense, this is obviously an incredibly important problem. And that is, if you look, this is the rate of infection worldwide. And the really bad places, the really tough ones, are sub-Saharan Africa, and even some of the places up here. These are incredibly bad in terms of infection rates. And you're talking about 30% of the adults in Africa could have some form of AIDS. And so this is an incredibly difficult problem for us, and obviously one that's really, really important. And if we look in the United States, this is child and adult deaths. And so as you look at this, there are obviously millions of deaths. This is a really problematic thing. The only good thing that you see from this, this graph is the fact that it's leveled off and is actually going down a bit. And it's actually going down substantially more over the next few years. The other thing that's good for us is that this is actually leveling off up here too. And these are people living with AIDS. So this is people who have the infection, but they haven't died, and they maintain their lives reasonably well over the time. Okay? Now, I found this one to be a really interesting graph. The reason being is, if you look at what's going on here, we've put together all the different types of deaths that are out there that are possible. So you have cancer, suicide, AIDS, stroke, heart disease, all these different ones. But the key is, look at the AIDS one, and look what's really important. If you look, it's going up and up and up. And then about 1994-95, look what happens. Suddenly now, this takes a huge drop in terms of the overall infections that are out there and the people that are dying from it. This is a really, really important step for us. And I want to show you what that comes from. Okay, so this was, in the final one that I was looking at, this is the rational drug approach. And that is that we knew there was an AIDS infection. We had a pretty good idea of what was causing it. We knew that there was some viral thing that was out there. And if it's a viral thing, we can attack it with certain drugs. We can do things to it. But we need to know what it looks like. We need to have an idea of what kind of enzymes are in there, where we can attack it, what we can do to it. And so what I'm showing you here is the reason this is considered to be rational drug design is because they didn't just take thousands of compounds and put them into people who had AIDS. What they actually did was they developed a drug that's never been seen on Earth. They developed something that fits into an enzyme and was developed through a computer-assisted drug discovery system. And that is, this is the crystal structure of the HIV protease. This is sort of the cartoon structure of what it looks like. But the key here is right in here, this active site. And essentially, these proteases, what they're doing is they will take, the AIDS virus has these proteases, and it will take all these different kinds of large proteins, and the proteases will chew them apart and spit out the pieces that it needs to run. And so what was thought about was, is that a reasonable target for some sort of drug that we can develop here? And they thought about it, and they said, okay, well, if we can do this, and this is where this thing is that's cleaving apart all these pieces that make up the next virus and move on to make more viruses out there, then what happens if we stop that? What if we put something in here that binds to it and won't let it do that? And they figured, okay, well, this is a reasonable approach to doing this. And what they found was the first drug that they looked at was something called saquinavir. Saquinavir was the first HIV protease inhibitor that went out there and actually fits perfectly into that structure. We'll see that in a couple minutes. But what I want to show you also is the three different areas of the AIDS virus that we can actually attack. And that is, this is sort of the life cycle that we go through. And this is that an infection occurs, and then we have to go through these different things. So reverse transcriptase, this one you may have heard, drugs like AZT is a reverse transcriptase drug. And so this one will go, and we can use those. They're very toxic to people. So you're killing the virus, but you're also killing the people over the course of this. Integrase has not been used very much. We haven't attacked this very much. We don't know that much about it. So this is something that's starting to be of interest to us. But the one that's more important to the ones that we're using these days are these proteases. And this is one of the areas that we attack with this. And so this is where we've developed drugs that are going into this. And so the most important drug out there is darunavir. It is the one that's most used now, and I just want to show you. Here is the HIV protease. Darunavir is here, and this is the structure for it. And the one I wanted you to see is if you look at all the blue and red here, those are important. Those are points of mutation within the HIV protease. Got it? So that HIV protease can mutate easily to all these things. And so it's very difficult to develop drugs that won't be gone in a short amount of time. Okay? Here's the saquinavir and darunavir. And the only real difference here is that this is considered to be a non-peptide. So this one was torn apart in the gut because of these peptides that are here. It did not last very long. But we can use darunavir, which is a non-peptidic, so we don't break all these bonds that are there. This, even though it looks like it, it's called a carbamate. It's not a peptide bond, so we can't break it as we normally would. So this darunavir is the second generation of these. And also, this part of it that you see here was really important. It helps bind. It helps make this bind about 1,000 times better than does that. Okay? And then the last part of this is here's darunavir, and then this is all the things around it that are binding with it. So just to show you that all the things around it actually cause some sort of bonding to things, hydrogen bonds in particular, to all these different areas, to hold it in there very tightly and keep it from anything else coming in there to be split apart as a protein. Okay? So that's the last part of that. And just to show you this real quickly, this is our approach to 4-ball. 4-ball is I call this a molecular contradiction because it is a tumor promoter, but it's also an anti-tumor compound in various forms. And so you would think of that, for most of you, you'd go, well, what do you want a tumor promoter for? But realistically, what that tells us is a pathway for a tumor to start. And so what we get out of that is we can figure out how that goes and attack somewhere in that, the formation of those tumors. And so 4-ball is of interest to us in terms of that. And then the last one here is, this is our approach to it. We've been able to take something that looks like this to make something that looks like that ultimately then turns into something that looks like 4-ball. So this is chemistry that comes from my lab to make molecules that look like this. And these retain a fair amount of activity in terms of 4-ball type activity, although we haven't tested as many of them as we would like. Hopefully I have a professor in North Dakota that will take a look at these and do some of the work with them. All right, and then finally, because I'm running pretty late anyway, I have had the joy of having an incredibly wonderful group of students, both undergraduates, graduates, and others. Over the last 24 years, it's been a joy for my life. And also, I have to give a big thanks to OU because they let me do something that I really love in my life. So, and I want to thank you guys for listening.
Thank you very much. Does anybody have a question?
Go ahead.
I missed this earlier, so I apologize.
That's all right.
Why is it that Taxall only targets cancer cells and not necessarily tissue?
What we find is that the forms of that tubulin tend to be more specific for specific types of tumors. And so there are markers on it that make it easier for the Taxall to bind to the beta tubulin of that. Now, there are a lot of interesting studies that have gone on where they've been able to put groups on there that help target different types of antigens and various things, so we can actually put it more specifically. And there's a lot of work that's gone on in the last 5 or 8 years in terms of being more target specific. Because obviously, as most cancer drugs are pretty tough. If you don't kill the person, you're lucky. They're fairly tough on most humans. The last. Sure, but generally microwave is part of what we do. I mean, it's a way. Oh, the question is, have I used something like microwaves in terms of doing a synthetic methodology? And the answer is yes. We use microwaves to cause reactions to occur in a better fashion. Sometimes ones that don't work very well, we can put them in to a microwave reactor, and they'll work better. Now, biologists and structural biologists do amazing things on their own, and they do them very differently from what we do. But there's a good partnership between the two of us to end up with drugs that are important in terms of helping some of these disease states. So, we try and, most of us are pretty open to a lot of different things. But as a synthetic chemist, I'm going to do something that's pretty standard, most likely. Yeah. That's okay. That's okay. Probably not. That's a good question. So I think what you're getting at is, will there ever be a cure for these types of cancers and things like that? Right now, if you look at AIDS in terms of that, we haven't cured it, but what we've really done is we've made it a manageable, you can take drugs daily and your life is not, it's not changed in any realistic way. You have to be careful and you have to make sure you take the drugs all the time, but it's possible. The things that are coming out that are interesting, though, are ones where they're giving, where they give, they're trying to figure out vaccines for cancer and things like that. There have been trials, and I saw recently there was one that they thought was working. Unfortunately, all the cases that they used it on, they were in remission. They're back to active cancers, though. So, do I think it's coming? I think at some point, but the problem is, this is a really tough problem. There are lots and lots of differentiations of the cancers themselves to the people that have them. This is why when women get breast cancer now, they usually do a lot of genetic testing on the exact cancer that you have because they want to direct the correct compound to use against it. So there's a whole lot of things that go on with that. But as a scientist, I always have hope that there are an incredible number of really smart people out there that work really hard. And so, yeah, I will die going down that these things will be taken care of.
You got a question right there.
Yeah. The 4-ball's been in our lab for quite a while, and we don't have any compounds that would be called a lead, which is what we're really looking for. Something that will that you would give to, we're small, and so I can't those running those trials, we can't do that. That's way beyond our thing. But what we hope is that somebody like Pfizer would come in and say, Dr. McMills, your compound is really neat. Here's $10 million. We'd like to buy that from you. And then they will move it along. Exactly. Exactly. So my child has to go to college next year, so it needs to be sooner than later. So. Ma'am, sorry. When I was an undergraduate, I actually met Professor Wender, who was the one who had done Taxall. His work, when he originally started, was in 4-ball, and I was really interested in it as a molecule. It's got really interesting pieces to it. That 7-member ring in the middle of it is really interesting to us, especially the way we approach those. We tend to be able to make 7-member rings fairly easily. And so that automatically struck me as something that was of interest. But it was also that it sort of acted in this contradictory manner in terms of how it was acting. And so that was, those sort of things pique your curiosity as a scientist. Go ahead.
Last question.
No, those have been done.
Well, my question remains, when those kinds of things are being done in the lab, what is done, what kind of time to assess, this is better?
This is the $64,000 question, and it's the expensive part. Oh, so the question is, with these tweaks, how do we know that they're doing anything better, worse, whatever? That was your question. And so normally what happens with these compounds is we have various types of assays that we use in terms of, the simple ones are going to be cell assays. So we'll be able to take the compound, put it in with various types of cells and see if it interacts with them, kills them, does not kill them, whatever goes on with that. And then we move into things like we'll give them to animals and see if it does the same kind of thing. So, you know, in a lot of cases, you'll have a mouse that has a solid tumor. Into that solid tumor, you'll place some analog and see what happens to it. Does the tumor shrink? Does the tumor grow? And so, you know, we don't do that until we have a pretty good idea that the drug itself is doing something reasonable. Nobody's trying to put an animal under really, you know, bad things. But usually it's cell assays, right? Yeah, yeah.
Okay, so with that, I'd like to thank Dr. McMills one more time.
Thank you.